Ladies and gentlemen, it's a great pleasure to welcome here a person that has done in nanomedicine a huge work, and outcome was 1995 Doxil as a medication which Chesi Barenholz has developed with Alberto Gabizon. For those who do not know, Professor Yerzekel Barenholz is professor at the Hebrew University and Hadassah Medical School in Jerusalem. And we have at Klinam was happy to have him for many years as a great, great speaker. Please, Chezi, your talk.
Okay. Thank you very much, Biet. Well, as I talked with Biet a few minutes ago, we know each other for 11 years, and we met in Klinam, of course. And since then, I don't think I missed any of the Klinam meetings, trying to help Biet as much as possible. I think it's a fantastic meeting. It's really heaven for anyone that interested in nanomedicine and the nanoscience related to it. So I think this meeting is really exceptional achievement because we all wonder how it will go, and it seems to go so well that I'm sure everyone enjoying it. Of course, we miss the kind of the face-to-face touch, the small talk, and this is really important. But as it said, the second best seems to work very well. So Biet, as usual, is doing exceptional good job, and he proved it again in this meeting. Thank you. I'm really want to thank you. I'm sure in the name of everyone, like I'm doing in every meeting, but every meeting I can see everyone, and now I see only you. So thank you very much, and we hope that the next meeting will be face-to-face, but we know even if not, we'll have another good virtual meeting. Thank you, Bia. Okay, actually, I'm not going to talk in Doxil. I'm not going to touch Doxil today. I was very excited to see the new generations that are coming, to see all the excellent studies that were presented in this conference so far, and I'm sure will be presented tomorrow. I'm the last one to talk today, so you know, in usual meeting we can say everyone is very tired, probably want to go home, but here everyone is at home anyway. So in this respect, it's easier for me. I'm going to talk on a major issue that actually people don't want to touch, and that's one of the problems. I'm going to talk about hospital bacterial infection and their prevention and some related topic. You know, we are now at the COVID-19, so there is no way you are not going to touch on it too, although I'll not focus on it because I think the other issue is of the resistant bacteria is even higher, even larger. So I have to share with you my conflict of interest. Actually, I'm emeritus professor now for, I think, something like 10 years. I still have a huge lab, not huge, but large lab of 12 people. I still have to worry together with all the companies for salaries for about 40 people all the time. So being formally retired doesn't mean much. Anyway, I'm going to talk about 3 projects, and that's why I'm going to discuss this conflict of interest. I'm a board member of Sonovia. It's a company that make now the masks. Here you can see the Sonovia mask, antiviral mask. I'm not going to talk much about the mask, but I'll talk about the Sonovia, the invention of Bar Ilan University, which was licensed by Sonovia. I'm also was one of the founders and a member of the board of a company called Polypid, and I'm going to talk about one of the few of their products that really help to prevent hospital bacterial infection. And finally, a project of my lab. I'm a head of the lab, still the head of the lab, and I'm co-inventor and co-development of a drug called nano-mupirocin, which I'll tell you about. This has some kind of things we learned from Doxil, but it's very, very different for me. Well, we are really at a kind of a time that we have to raise an alarm. And the reason is, if you look today, cause number one for death, actually today, there is a competition between cancer and heart diseases, but it seems that in most places, cancer is now number one. And AMR, which is antimicrobial resistance, have about 10% death out of it. Still very large number, but if we wait longer without doing much, what we can see that the situation will be shifted, and the AMR, the issue of the bacterial resistance, will be number one cause of death in the world. And that's why the agencies, different agencies like FDA, especially FDA, to lesser extent EMA, really do things in order to convince industry to go and deal with this project, but the response is very, very poor. For various reasons, there is very small focus on this. And actually just 3 days ago, if I remember, or actually on October 22nd, 4 days ago, there was a paper in Nature. They say no money for new drug, they aim for antibiotics. And they said that actually they give all the reason, the justification that company bring in order not to develop antibiotics. I don't want to go into it, but this really frightening to read it, because it just shows us that the main things behind everything, the drug development is really money. And that's really a very, very sad and very bad message. Here you can see the implication of it. If we look on the really what happened through the years, we can see that at the 1890s, there were a lot of antibiotics and very little cancer drug developed. But it became even worse today. Here I have it only to 214, but the situation even worse. You see that cancer is taking over by far, and very little antibiotics is really developed. And that's really a major, major threat to the health everywhere in the world. What are the ways that we can deal with this, with the AMR issue? So I kind of see 3 different ways. First is a passive way, but which we take much better care on patient environment in the hospital. And by this, we can reduce the chances of the patient to be infected. I'll talk about this. The second is take an active measure by patient pre-treatment. They are already in the hospitals and they go surgery, and there is a very high percentage of infections. Some hospitals, really, I checked through the world, they have 30-40% chances that the patient that get in and through a surgery will get infected with bacteria. Many cases, they are resistant bacteria. So can you take some active measure to prevent it, like pre-treatment of the patient? I'm going to talk about it in relation to surgical site infection, or what we call in the field the SSIs. And the third one is the most complex one, is developing a new drug product to treat AMR. I'm going to talk about these three topics. Actually, because of this and because of the pressure from public and from WHO and the CNC and the CDC, the major company gathered together and built a fund, and they came with a statement that's the statement. There is a need to develop antibiotics, and actually you can see something very strange that never happened again. What's the problem with resistance is if we use too much the antibiotics, after a while we get resistant to it, and then we are out of these antibiotics. It will not be helpful against the resistant bacteria anymore. So the idea is to build a very different concept in which the governments or this fund will encourage the companies to make new antibiotics, and you pay for their potential use, but not for their use, and then they will be served only for the special cases that we need them. So the company get money for not using the drug actually. So it's a very different economic model. You can see it again in this Nature paper, and there are a lot of other publications about it. This is a completely new idea that came because otherwise no one will really put any money to develop a new effective antibiotics. Let's start with the first one, which is taking better care of the patient environment, reducing the chances of patient infected. I'm going to share with you, it's not my work, it's a work of Professor Aaron Gedanken from Bar-Ilan University, technology that the university licensed to Sonovia, and actually the masks here are based on this technology. It's antiviral and antibacterial. I'm going to talk more about the antibacterial, and we talk about the kind of the mechanism. So it's really classical nanotechnology technique. You can see the nanoparticles. They are small nanoparticles. You can see it on the cloth, on the cotton cloth, on the fiber of the cotton. You can see many nanoparticles. In this case, it's copper oxide, but the company is using zinc oxide, because copper oxide has a kind of color that no one wants, the color of copper that reminds things that people don't like to see. So the zinc oxide now is replacing, and they made it by exposure of the cloth in a special machine that was built through support of the EU grant, that can, while sonicated, make the zinc oxide particles, nanoparticles, below 100 nanometer particle that stick in a very strong way, that even if you wash with a washing machine for 60 or 50 times, the power remain, and you can see their effect on preventing the bacterial infection remain the same with the after the wash. So I think it's a very nice invention, a very important invention. Can have a lot of applications. I don't want to go into it, but the mechanism is known for quite some years. What we know is that zinc oxide, the metal oxide, that damage the bacterial and the viral membrane. We all know that the COVID virus actually has a membrane, and that's why you can kill it with alcohol and with detergent, but actually also zinc oxide destroyed this membrane. Then it permeates. There is some papers show it make hydrogen peroxide that really destroyed the protein there. There are no full proof yet to the hydrogen oxide, but there is a proof to the destruction of the membrane. Also, we know that the zinc ion that released in a very small amount as really can bind very avidly to biomolecules like protein, nucleic acid, and carbohydrate, and destroy them also. So there are few mechanisms, and there is a reference actually here that you can look if you're interested more in the mechanism. One of the experiments that was done is to take a cloth, actually to coat it to a textile to make from it all the things used in the hospital, like the sheets, the blanket, the dresses of the patient, the dresses of the medical staff, and really make a clinical trial that was done in Bulgaria. Since then, there are more trials, but this has all the full data, and you can see very clear the impact different days. You can see that without this, the rate of infection of the patient that was measured is much higher than if they use these special dresses, special coated. So you can reduce the level of infection. Actually, this mask was tested for antiviral against the COVID. They have a very, very good impact. Was really started by accredited lab, and you can see 99.34% of the virus really are destroyed, and there is also safety, good safety to it. Okay, I want to move to the second part, and this is the active measure by patient pre-treatment to prevent surgical site infection. So you can see CDC, WHO data, you can see the number of infection, tremendous number of infection. You can see number of deaths in the US alone here, and a lot of number of complication. You know, hospital in the US now pay punishment if there is infection with such bacteria. So there is a big pressure now, a big motivation try to reduce, and that's what we in Polypid took advantage of, trying to convince hospital really to go for this technology. Clinical trial, I'll show you data. Phase 2, we have now phase 3 data, but I cannot show them yet. So actually, it's used in broad types of application, like indication, like osteomyelitis, and the results are positive. First, we did a lot of compassionate data. I think the idea of compassionate is not very good because it's not controlled, and you don't have the placebo, and therefore, but now we have a good clinical data. And I show you really the effect, the impact of this. So first, the burden of the SSI, you can see it increased, estimated percentage is very high, 30% in colorectal surgery, 20% in healthcare on the average, and you can see how many days you added to the treatment, not talking about the death that I showed you before, and the expenses related to it, you know, in US, $10 billion yearly, and in the EU, 11 billion euro. So it's a huge, huge burden on the economy. Okay, what happened when we do surgery, not me, of course, but the physician, you know, whatever they do, it's very difficult to prevent the infection. In the best hospital, you can look at the number, there are somewhere between 10 to 15% of infection. Actually, I have a project together with Cleveland Clinics, and they told me, and with a surgeon, dealing with breast cancer, and he told me that what they did in Cleveland Clinic, they checked the air in the operation room, and they did and they actually test the level of bacteria there, with a DNA test. So it's very easy to know it's bacteria and even know which bacteria is this. And they can never get a clean air that is free of DNA of bacteria. So it's been very difficult to deal with it. Some hospitals I look through the world, they have 30, 40% infection, post-surgery infection, very bad. So today, the bacteria is going in. So today, what people are doing really is pre-treat with systemic antibiotics. Almost in every surgery, there is a pre-treatment with antibiotics, but it seems not to be good enough because still we have a lot of infection. And that's why the idea is to give a local treatment at the time of the surgery, at the spot of the surgery. And the idea is that by this, we can get very high concentration at the site. And you know, all the issue about resistance is a issue that the minimal inhibitory concentration, the MIC, was shifted to a higher dose. But if we give very high local dose, we can overcome and override the resistant issue, which we demonstrate in polypid clinical trial. So that's actually the idea. So what you do, I'll show you actually what we do. We have the paste of the drug and it's spread on the site of the surgery, of the operation of the surgery before the surgeon really closed the site. And then you still give systemic antibiotics because you have to, but it seems that this is really doing the job very nicely, as I showed you. So we have different drugs. One of them we called Diplex 100, and this was directly made to prevent the SSIs. And we can see that we get very high concentration at the site. I'll show you. One administration, you don't open it anymore. You just close the site. We know that the drug is released for a period for 3 to 4 weeks. We can direct it. I'll show you how we are doing it. We can control the time. And the concentration is all the time zero order, very high, sufficient. The free drug is sufficient level because what the drug that killed the bacteria is the free drug, sufficient level, much above the MIC, so we can overcome all the resistance. We use doxycycline, which is a broad spectrum antibiotic, so it's work against really broad spectrum of bacteria, including staph and MRSA. The administration is very simple. I don't have time, but we have a movie to show it. We have a movie that we made for physicians, very easy to do. And the patient compliance is very good, and the recovery is faster, as I show you in the rate. So the secret is getting effective concentration of the drug for 3 to 4 weeks on the site that will do the job. Okay, here just show you how it works and actually what happened. What is the drug? I'll go into detail more, but basically we have particles that are quite large, but they are coated with layers of phospholipid, cholesterol, and a polymer. We can work with different polymer. We can work with different phospholipid, and this allowed us to get different rate of release. And actually, through this, the alternate layer allowed to have drugs either associated with the polymer or associated with the lipid or in between. So actually broad spectrum of drug, we did nucleic acid, we did protein, we did peptides, we did antibiotic, we did steroids. All of them works well, and the company has patents on all of them. But I'm going to show with you the antibiotics because this is the most advanced, and also the clinical data are very, very good. So the plex protect the drugs all the time. Stability is extremely good. And what happened when it come in contact with the water or with the fluid, interstitial fluid at the side, there is peeling of layers, and then drug is released slowly. And we can make any number of layers we want. So, and for different materials, so we have a very good control over the release rate. So it's on site and very simple procedure, although it's not invasive because the patient undergo surgery anyway. So you just add it at the end of the surgery. Here you can see from our paper, the 212 paper in JCR, they describe the system, and you can see how it built the alternate layers of polymers, phospholipid drug, phospholipid polymer, and so on. And that's actually the kind of the structure of the particles. We did X-ray study on it, and you can see that only when you have mixed of all the component, in this case is DPPC, cholesterol, and the polymer, only then you get a very clear structure. All the rest have a much poorer structure if you miss some of the components. So it's really very important to get all the component to get the structure. Just giving you some idea about the size, which is really not matter very much. What matter is the kind of the nanostructure of what happened on this hard sphere that is coated with the layers of the material. And here you can see how the release rate of the drug, which is all zero order, affected by the lipid. If we have DMPC alone, we get 80% release in 11 days. But if we have DSPC alone, DSPC alone, C18, the rate is doubled. So we can control it to the level we want in this frame. And that's very nice. Also zero order, you can see that if you don't have the whole component, you get a kind of a complex kinetics, very, very fast. But if we get all the component, this is a formulation, I think based on DPPC, you can get a kind of a zero order release, which ensure a kind of constant amount of drug at the site for a very long time. And actually this expressed in the way that we can see how the MIC of the bacteria change. This showed the stability impact. If we give this, you can see the stability remain all the time. But if you give the drug alone, you can see MIC is keep increasing, which means the stability of the drug is reduced. So actually, if we look at the amount that we have to give, you can see that at the site, we give something in the range of one administration, 55 to 164 milligram. That is, if we give the equivalent treatment is done with systemic treatment, we need 60 pills, and the total amount is 6,000 milligrams. You can do the calculation itself. What's the difference in the exposure in terms of toxicity and all this aspect. And you can see that if we measure the amount of the drug at the site of the infection, the free drug, you can see that it's constant all the time. You can see for a very long time. And here what you see in the plasma, you can see this is free drug going down very fast, very high concentration. And this is what we see in the plasma of the patient when we give the diplex. You see a very, very low amount, much, much below the toxic level. So reaching the other organs, the amount that is available is very, very low. And that's one of the phase 2 clinical trial. And you can see here the advantages. This is the shortening of time of the treatment from 267 days to 162 days, very significant. This is in case of osteomyelitis. And you can see here the impact and the shortening of the time if you look at the different parameters, and you can see that, for example, in terms of pain, there are almost no pain in the case of this drug, of our duplex, while there is a lot of pain in the case of the free drug. So many signs show the advantages of this drug. Okay. So we are now in two phase 3 clinical trial with the duplex, one in soft tissue, the other in bones, and there are quite few patients in them, and these are long studies. It will take long time, and actually FDA really encourages, gave us a lot of discount in this. They really need it, and hopefully in about a year or so, in the next meeting, I can present you data of the phase 3 clinical trial. Hopefully they will be good and we can get the drug approved. The company, based on this data, actually went public and IPO was very successful, just during the COVID time, actually. The third product is not that advanced. It's something developed in my lab by my team, and this is the nano-mupirocin project. And you can see actually the liposome of nano-mupirocin. There are 80 nanometer liposome attaching to really adsorb to the bacteria. We know that they will go in and kill the bacteria. And actually, one of the unique things in this case, that the liposome with the drug work as intact system. The drug doesn't need to be released in order to kill the bacteria. Although if drug released, it also will kill the bacteria. And I show you proof to it. That's a very interesting. Okay, so let's, first of all, I have to admit that I myself was never thought I will deal with antibiotic in my lab. And by serendipity, it happened. I have a student now, she's running the project, Dr. Ahuva Cern, and she was my PhD student, and her job was to develop algorithm that can calculate by machine learning and artificial intelligence which are the best drugs to be used with liposomes. And she did it. There is a paper published, few papers, the last one highly cited is JCR for 217, you can find it. And one of the molecules she found was mupirocin. I didn't know what the molecule is. She did some study to show that it did what the model suggests is really true, and it can be loaded very nicely and in a stable way into the liposomes. Then we look at it, it was antibiotics, and I told Ahuva, forget about it, I'm not interested to develop it further. But someone came to us and said, Chezi, I mean, you are at the position that you should do something to support the tough situation that is today. You already made your share with Oxil, and he convinced me to go for it. So we work on this project, and actually mupirocin is antibiotic, which is amphipathic weak acid, and therefore it can be remote loading by one of the patents we have. We're using calcium acetate gradient. You can see the molecule, you can see the fatty part of it is a fatty acid of 9 carbon atom, and you can see the head group of this, that is the acidic group here. So it's, and we also, on the top of the regular loading, we added some other novel aspect, which is a way to control better the release of the drug. And the drug is entrapped inside the liposome by the calcium acetate, but it may complex with cyclodextrin with HPCD that control the rate of release. Mechanism of action is reversible binding to a very unique isoleucyl tRNA synthase, and it inhibits specifically protein synthesis of certain bacteria, not all of the bacteria. I'll show you which one it's doing the job. And this is very unique mode of action, not shared by any other antibiotic. So there is no cross-resistant with any of the antibiotic we know today. And as I show you later, it's very difficult to make resistance against this bacteria. Don't have time to show you the development, but basically we did with remote loading, we succeeded to load very high concentration of drug inside the liposome. It also made a certain structure, not the nice crystal, but other type of structure inside the liposome. and the high concentration enabled it to be used in human. I'll show you work in few models to show you the efficacy. It has superior PK profile. It's a pegylated liposome. We check few composition, and the best one we found is actually, we were lucky, was the doxyl composition, so we make it easier with the FDA. As I told you, the liposome itself with the drug, not without drug, kill the bacteria directly. And what nice is that it has a very high therapeutic index because the NOEL of the drug is huge, 100 milligram per kilogram, which is much, much, much above the concentration you use for the clinical trial. Actually, that's a kind of a statement which the EU and other bodies said what is a novelty for antimicrobials. So absence of known cross resistance because of the AMR issue. It has to be new class, new target, and new mode of action. Today, there are few new drugs that are in development, but only 6 of them show one of these parameters, and one only show the 4 criteria, and we also show the 4 criteria in our nanomupirocin. So that's very encouraging. Let's look at some model. This is a necrotizing fasciitis model. It's a very deadly disease. It's not of high interest because the number of people that the number of people that are infected is very small, but the disease is skin disease. It can lead to death basically. And actually what we did, we work with a team in Hadassah that have the model. In all our work with different teams. This is Alon Moses' team. The head of, at that time was the head of infectious diseases at Hadassah. And what we did, we treated it with the nanomupirocin and different doses, injected IV, because the main things of the drug, which I forgot to tell you and it's very important, is that the drug can be used in a cream, but it cannot be injected because once injected, it destroys in a matter of really few minutes and bind to protein that also inactivate it. So actually, although people will try different way to make it a kind of for parental use, there was no success on it. Actually, it was impossible to get any work. So the drug can be used only in a cream, and it's quite effective in a cream, but it cannot be injected. The change we made is that we converted it to a drug that can be now injected parenterally. And actually, that's a very successful, because what we can see here, that while if we don't treat, all the animal died, you can see quite fast. And this is different, free drug doesn't work. And if we have a low level, you can see we do some good, but with a higher level, we actually succeed to really protect all the animals. We also have movie to show that are in a good shape, they didn't lose weight and so on. but I don't have time to show it because I want to show you other models. This is a model of osteomyelitis. Again, first thing we want to show that actually the drug can reach the site, the tibia, where we have the disease. Also the kidney, which is a very important site of infection. You can see both a very high number of a very high level of drug there, much above the MIC. And you can see that we successfully reduced the burden. Free drug doesn't do much, a little bit. This is the control, and that's what we got with the nanomupirocin. And actually, you can see also in terms of the inflammation that when we give, while in the blank and with free drug, there is a very high level of inflammation, as can be seen from the level of interleukin 6. We reduce it almost to zero with the nanomupirocin. And this is the endocarditis model. Again, I don't want to make you tired with successes, but you can see here again that most of the animals survived, while with the free drug, there is no survival in endocarditis. And this is a MRSA in neutropenic mouse lung infection model, very problematic disease. And actually, it was as effective as vancomycin, a little bit more actually, but there is a lot of side effect to vancomycin, and there is a resistance that we don't have in this case. And you can see that the efficacy was very, very good. Look at the log of the number of bacteria. We reduce it dramatically, already four logs, which is very, very good.
5 minutes.
Okay, I'm close to the end. The interesting story is that, as I told you, we can see that nanomupirocin, as the nanomupirocin really interact with the bacteria directly. And if we put in plasma that mupirocin is destroyed, nanomupirocin works. And we can see it here. And there is also, we can see that the accumulation of the drug in the bacteria, while it's very, very low when you give free drug, and that's not surprise, but it's very, very much higher, again, much above the MIC when you give it as a nanomupirocin. Not only this, the liposome are taken up by macrophages with the drug, and we know the macrophages are serving as a shelter for the bacteria, and you can, with free drug, you can really kill the bacteria in the plasma, but then after you do it, after a while, bacteria in the macrophages are actually multiply, and then there is enough and they burst and you get a repetitive infection. So what we did here, we demonstrate in infection case that when we take the macrophages from the animal that was infected, and we can show that actually, that first there is mupirocin there, I'll show you in a minute, but the other thing we can see that we can kill the bacteria very nicely with these particles. So we also kill bacteria that are hidden inside the macrophages. And this show you actually a short movie that will show you the uptake of the nanomupirocin and with the bacteria inside the macrophages, and it killed the bacteria inside. So we have a very good toxicology. Actually, NOL is very high in all level. Pharmacokinetics, very interesting. 3 doses, no ABC at all. You can see we did 6 injections, and there is no difference between the first and the second in all the doses, male and female, very good. What's very, very important is IM dose. Well, you can see that it's much above the MIC. That's very important. And here it's again, we focus it now on gonorrhea. That's the application, the first indication we want to go for. And you can see this is the they actually male, female, and you can see that the pharmacokinetics is very favorable, much, much above the MIC, and really stay there for a very long time. You can see 40 hours or so, which is much above the time needed to kill the bacteria. And so toxicology conclusion, let's ignore it. I want to focus, and by this end the lecture, on the gonorrhea indication. It's a major issue. People don't like to talk about it too much because it's a sex-related disease. You can see 1.1 million cases, new year cases every year in the US, about 500,000 resistant bacteria, very major problem. Also relatively easy to treat because you need to give 1 or 2 injections. If it's do the job, the patient will be cured. So also in Europe, we have problem. In China, we have problem. So it's a very major market, and we want to go for it. So the first thing we have to do, let's just show you a study done by a CRO to demonstrate that actually it's very difficult to form resistance against this bacteria. And I want to show you that actually if we want to treat gonorrhea, we have to reach the vagina and the sex, for example. So we did an experiment to show that the liposome reached the vagina and also the drug. And what we did, we labeled the liposome with lysamine rhodamine PE, and we can demonstrate actually here that we get the liposome. This is we swab, we took some of the mucus, and we found bacteria with nanomupirocin there, which is a very good sign. And then we measure the quantity in these swabs. We measure the quantity of the antibiotics, and you can see the plasma of the patient, of the mice, and you can see the level of the swabs. Very high level, much above the MIC, more than 100-fold above, and actually for a very long time. Again, it's time to kill. And actually, there is no good animal model, so we are now planning a clinical trial in gonorrhea. I hope I convince you that that's a good direction. And actually, Dr. Caroline Dill, which is the chief of the enteric and sexually transmitted disease in the NIH, we have discussion with her, and she was quite interested in the project. We're discussing it. Maybe they will help us with a clinical trial because, you know, we need a lot of patients, and in the US, they have access to many patients. So basically, although there is no money for new drug, in case of the nanomupirocin, we already overcome the risky part of the development. We having good preclinical data. The other thing is very important to say that while in cancer, the difference between animal models and actually human are very large. In the case of antibiotic in general, the relevance of the animal models is 10 times better than it is in cancer. So there is a good motivation to develop, and I hope that we'll do it. And this is, I'm going to really present you Dr. Ahuva Seren. She's a great lady and really it's really doing a great job she's running the project. And thank you very much for your patience bearing with me at that late time of the day.
Thank you very much, Chezi, for this talk. There are several questions which I would like you to answer, maybe in short. The first question was where this article is. I can say it myself. It's in the issue 550 of Nature. The volume is 586, and it was published on the 22nd of October of this year. The next question is that there are you talked about this new business model, and
Yeah.
we would like to know, or is this model already in establishment?
Yes.
How long
Yes.
will it take before it starts? And are the states, you said this shall be paid in the new business model by states. Are the
Oh,
states
well, no, no,
prepared?
no. There is a fund. There is a big fund, $1 billion fund, already obligated by the companies and the government. Okay, so 1 billion. This is the first hit of this fund, and this fund is dedicated for the development and also for payment to the company for holding, not to really use it in a free way, but use it in a very controlled way, whatever developed. So first you use whatever available now, and then in the cases that need it, you will use it. You don't pay for the specific use, but you pay like a member fee to join this group, basically. So by
Okay.
this, the company that developed this drug will already get continuous payment all the time. That's the idea.
Will
You
these
pay like a
companies
member
also
fee.
will these companies also develop new drugs?
Yes.
The
Everyone that
question is
developed,
the question here is you have said that there are six antibiotics uh, beyond
six in development. Yeah, yeah.
included polypid and mupirocin.
Sorry, I didn't hear the
Is
mupirocin
that
is
included
no,
your two
no,
drugs?
no,
No.
no, 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 no,
So
not yet, not
for
yet.
only 8 drugs.
Yeah, yeah, that's right, that's right. It's no one wants
Will
to. You know, I was in many companies, and the science. I can tell you the science people like it. They want to vote. Then came the marketing people, and they really veto it. They really veto they say, no way we'll go into it. You know, let's say gonorrhea, you give 1 or 2 injections to a patient, okay? Well, some of the companies said, we want you for cancer drugs. Come to us with cancer drug, we get you money, okay? We give you money. But for antibiotics, our shareholder will not like it. It's terrible. It's terrible.
Okay, and a last question. What value could phage therapy have in the context of antimicrobial?
Say it again, I didn't listen. What?
What could phages therapy have as a value within antimicrobial?
Okay. There is a big argument about it. There is a big argument. I mean, the problem with the phage is on one end, it looked like very promising. And on the other end, you know, the worry is that you develop something and it has to be very specific. And if there is some changes, then it will not apply anymore. The kind of let's say the versatility or the kind of degree of specificity of the antibiotics is much less than the specificity of the phage. So they're worried about the phage, that already there are cases that you develop a phage and then the bacteria is a slightly different one and the phage doesn't do anything to it. And
Okay,
that's why, in a way, not necessarily antibiotics by the classical meaning, but something on this meaning that's a little bit broader perspective, broader use will be much better. And also there is indication that the bacteria can be resistant to phages too. So it's the bacteria are really unbelievable. You know, they are one of the best God creation, unfortunately, but that's the case.
Chezi, thank you so much that you took so much time to speak to us today. And we are
Thank
very
you, Glenn.
glad.
Thank you.
We are very glad
We'll
that
see
we
you tomorrow.
heard you. And we see each other soon. Yeah,
Yeah.
tomorrow. Great. Thanks a
Bye.
lot. Bye.